Welcome to Lecture 9, and we're going to talk about cycloalkanes today. We'll talk about the conformations that are most energetically favorable. So our last lecture covered the Pogel exercise 5A, and it was the conformations of kind of straight chain alkanes. And you learned how to draw a Newman projections. So let's just do a little review to see if we can name a Newman projection. So I'm going to give you a Newman projection, and we can convert that to a name, because you also learned how to name alkanes. And then today we're going to just talk about the cycloalkanes. And I want you to learn how to draw the cyclohexane chair conformation. So that'll be something that you'll see on your exam for open response and your quiz. And so you want to draw those, and then you're going to have to predict the more stable di-substituted chair conformation. And we'll do that through Pogel 5B. So we're going to do all those things today. Okay, so let's just do a review here. Let's see if you can name this compound. You want to be able to take a Newman projection, and you want to convert this to the Lewis structure, or the condensed. Okay, so if you look at this Newman projection, is that, okay, so you have staggered, or you can have eclipse. Which one is it? Okay, yes, so it is staggered, and in staggered you can have an anti, or you can have gauche. Okay, and which one's the lowest energy? Yes, so this is your lowest energy. And do you see your bulky groups here? Your bulkiest groups there are dihedral angle 180 degrees. So this is anti. So this would be your most stable conformation. Now, to convert this name, okay, so we have CH3, CH2. You go here, and then you got this carbon here. So that's that carbon, and you see how it has two hydrogens? You follow these lines. So that's CH2, and then you got to go to the back carbon. So this back carbon's here, okay, and that has two hydrogens. So that's CH2, and then you go to this one here, CH, and then CH3, 2. Now, if you want to, you can also go ahead and draw the Lewis structure, so you can name. Because sometimes the branching can make it hard for naming. But you want to take your time, and you want to make sure that you do the condensed formula. You need to get the structure out of the Newman. Okay, so let's just make sure, let's check our carbons. We have the red ones there, we have the blue here, we have the green here, and then we have this CH, and then we have the CH3 and the CH3. Now, in our naming, we find our longest chain, right? So 1, 2, 3, 4, 5, 6. So what's 6? Hexane, right? And then we need to number to get, well, where's our substituent? Our substituent's here, right? And then to give it its lowest number, you go 1, 2, 3, 4, 5, 6. So that would be 2-methyl hexane. Okay? And so one of the things you need to be able to do after doing lecture 8 is to take these Newman projections, convert them to the condensed formula, and then be able to name them. All right, so now before we leave the straight chain alkanes and go on to the cyclo compounds, the reason why we do zigzag, okay, for our compounds is because the higher alkanes, this puts the methyl groups here, this whole big group here, and this whole group in the anti, okay? So if you do that, you'll see that that already puts it in the anti-gauge configurations by doing the zigzag. All right, so now we're going to do cycloalkanes. So what is the smallest cycloalkane there is? That would be the 3-membered, right? So I'm on page 166 in your textbook. And so we're going to look at cyclopropane. And see if you can name all of these. We're going to list, this is the hardest one to draw because that's not right. I'm trying to draw a 7-membered ring. It's easy to draw more of a hexagon. 1, 2, 3, 4, 5, 6, 7. Okay, there's 7. Okay, so we'll do those. See if you can name them. Okay, so we got Cyclo Butane, Cyclo Pentane, Cyclohexane, Cycloheptane. Okay, so that's the nomenclature. Now, if you remember, we talked about a compound that looked like this when we were doing isomers. We also talked about this. Okay, and because this has a double bond, it has cis-trans. So these have the same molecular formula, and they have the same connectivity. Okay, so if they were different connectivity, they would be constitutional isomers, but they differ in their arrangement in space. And so this makes them stereoisomers. And so when we said so this is 1, 2, 3, 4. This compound is 2, 3 difluorobutene, ENE. And if you look at this one here, this is 2, 3 difluorobutene. See the ENE for the alkene. All right. And usually you would put, before that, you would even put insert here. So you'd want 2, 3 difluorobut, and then you would want to say 2-ene because you have the double bond on the 2. 2, 3 difluorobut 2-ene because the double bond's on the 2 carbon. Now, but these are different. These are stereoisomers. So what you have to do is if you have the fluorine on opposite side, that's trans. And if the fluorine on the same side, it's cis. So to separate this, you would write trans before that name, and this you would make cis. And so these are stereoisomers. because you don't have freedom of rotation around that carbon-carbon double bond. Now the reason why I bring this up is because in the cycloalkanes you have restricted rotation. So we're also going to have to look and name these cis and trans. We're going to have cis and trans isomers. And anytime you're putting the word cis and trans isomers, these are stereoisomers. Okay, so we're going to go through here and we're going to talk about the different properties and describe these cycloalkanes. Okay, so an example real quick before we leave the cis-trans. So like if you have a CH3 here and a CH3 there, what would this be? 1, 2, 3. This would be called trans dash 1, 2 dimethyl cyclopropane. Okay, and this one here would be cis 1, 2 dimethyl cyclopropane. So that's where you capture the cis and the trans. Trans means they're on opposite sides of the molecule. Cis means they're on the same side of the molecule. So you have to be aware of that with these cyclo compounds. And one other thing before we leave this, you can also see it written like this. So if you have this compound here and you do something like this, you can do it with dashes and wedges, which is a two-dimensional. In this case, this would be trans dash 1, 2 dimethyl cyclopentane. So be sure to know how to name those. Review that in your book. Okay, so the two things we're going to really be talking about with these different cycloalkanes is a thing called angle strain. And if we were in classroom, we would make a model, okay, with the model. Okay, this is also called Bayer strain for the professor that came up with Bayer strain and described it. And then there's ring strain. Okay, so you need to know the definitions between the two and then how they apply to these different rings. So when you have these compounds, let's look at this cyclopropane. So you have a hydrogen that goes up here and a hydrogen goes down there. What is the hybridization of that carbon? sp3, correct? So that carbon is sp3. So that means its bond angles are 109.5 degrees, right? Now, is that possible? What do you think this bond angle is here? That bond angle in a cyclopropane is about 60 degrees, okay? So that's called angle, that would be angle strain, okay, because that's the strain of the angle. So this is 60 degrees, and that's strain, okay. It wants to be 109.5 degrees in each one of these, because these are all sp3. All right, so that is ring strain. Angle strain. Let's look at cyclobutane. So you have a hydrogen here and a hydrogen there. That carbon, sp3, what do you think that angle is? That angle is about 90 degrees. Okay, so you have angle strain. This is angle strain. It's the deviation from the bond angles of 109.5 degrees. Okay, let's look at cyclopentane. Cyclopentane is what is the bond angle? Cyclopentane 108. So why did I put Celsius? 108 degrees. Okay, so that's not bad. All right. And then when you finally get to cyclohexane, you have 109.5. Yay. Okay. So you'll see because of this, 5 and 6 membered rings are special. Okay. And you'll find in organic chemistry that 5 and 6 membered rings, if they can be made, they will be made in a reaction. Okay. And a lot of it's because these ring strains are pretty good. Okay. And then in cycloheptane, we don't have any problems with um, the 109.5. The problem is sometimes you have too much entropy. It's like they're flappy and they're big, and to make them, to get them to come together, it's not ideal. But these 5 and 6 membered rings, these are ideal. And so you'll see a lot of 5 and 6 membered rings in nature. So that's angle strain. The other thing that you have to consider, the other parameter, is ring strain. And so what is ring strain? Ring strain is your Newman projections. Okay, so when we go back to the Newman projections, what are we really looking at Newman projections for? You're looking for the dihedral angle. Okay, and you're looking at sterics. Okay, these are sterics, and you're looking at whether you have gauche and anti interactions, or do you have eclipsed? Okay, conformation. So when you look at cyclopropane and you put all your hydrogens in here, right? And your eyeball looks down this carbon here, this red carbon, what's happening here? You see those hydrogens? They're eclipsed. Okay, and that's not energetically favorable. Okay, and so this is called ring strain. This is called torsional strain. Because, you know, we can't just give you one name for things like ring strain. They also have to give you torsional. And so it will actually bend these bonds. So these bonds get bent, and just to avoid this eclipse, so you just note that you do have a lot of torsional strain and they're eclipsed. Cyclobutane will form a conformation called slightly folded. Okay, so cyclobutane will go to a slightly folded. Okay, so these are names. You can say, okay, the cyclobutane is in the slightly folded conformation. And the reason why is because if it folds this a little bit, then you can relieve this. And so these are total eclipsed. Okay, there's no getting around it with cyclopropane, but this becomes just eclipsed. Okay, so these become eclipsed, but not total eclipsed. Okay, and then cyclopentane, it actually goes to this conformation called the envelope. Okay, does envelope spell, was it an E? I never know. Is it an E? I don't think so. It's an envelope. Okay, so this portion here will flap up, and you might want to look at the textbook to see some of these in your slides. Your envelope has an E. Okay, you all probably knew that. Okay, so the envelope conformation will do that, and the reason why it does that is because then it can make sure that now you have no eclipsed hydrogens. Okay, so this is another reason why these are ideal. Your cyclopentane is pretty good, 5-membered rings. Okay, and then the cyclohexane, we're gonna look at energy diagrams. So I'm going to start a new slide for energy diagrams, and we're going to look at 3 conformations. And these conformations are all about the ring strain. So we're going to look at how to get these different conformations for cyclohexane. We're going to learn how to draw these chair conformations. So we're going to focus on that. And we won't even worry about the cycloheptane. You don't really focus on that. And so know these two things, angle strain and ring strain. Okay, so I said that we're going to look at the six-membered ring. So cyclohexane. So we're going to look at this. We're going to learn how to draw the most stable conformation. And I'll just tell you the most stable conformation is called the chair. Okay, so we're going to learn how to draw the chair conformation, which is your most stable conformation. And we're going to practice drawing that because you're going to have to do that on your exam for the open response. And then we're going to look at if you have substitution. So if you have a disubstituted chair cyclohexane, we're going to look at how to predict those, the disubstitution, with two substituents on there. And so we're going to look at some cis and some trans disubstituted cyclohexanes. And then after we do that, we're going to do our PG5B. PG5B. Okay, so that's our goal. So first of all, on page 173 in your text, you can also look in your slides, you have an energy diagram. And this energy diagram is an important energy diagram because it shows you the different conformations of cyclohexane. I'm going to try to get these heights. Okay. So these heights should be the same. And we have a little energy height here. We have some energy heights here. This should be the same. And then this one, and obviously I'm going to have to redraw that a little bit. Okay, so these conformations, the lowest conformation is the chair. Okay, so this one is the chair, and you're going to learn how to draw this. Believe it or not, that's actually kind of challenging. Okay, and then this chair looks like this. Okay, so this is the chair. And after I do this energy diagram, I will show you how to draw the chair. Okay, and label the chair. So we'll learn how to draw and label the chair. Okay, so at the top here, you have a half chair. This is the half chair. You don't have to draw the half chair. But I just want to show you that these go through the different conformations. You have a boat here. This is called the boat. And then here you have a twist boat. And here you also have a twist boat. Okay, so both those are twist boats. And you get energies. Okay, this is showing you that this is 23 kilojoules. Okay, the boat to the chair, this energy is 29 kilojoules. And this energy, the highest energy to the chair, the most stable, is 42 kilojoules. Okay, so what does that mean? That means at room temperature, the chair can interconvert. Okay, so this is equilibrium arrows, and they convert. So this part that's down, you see how this part is down, this carbon is down, and this carbon is up. Well, now this carbon is down, and this carbon is up. Okay, it doesn't mean that much when you don't have any substitution, any methyls or ethyls or isopropyls on that chair. But it does make a difference when you start having substitution. Now, why is another reason why the chair is very important? I don't know if you've ever seen this compound here. See if, let me just redraw this. Okay. Okay. See if you know what this is. Anybody know what this structure is? C6H12O6? Yes, that's glucose. Okay. You see how glucose is a chair with an oxygen in it. All right. Sugars. This is big deal. This is biochemistry here when you get into all the sugars. Okay. They go through this six-membered ring. The five-membered rings, if you look at those, what is this sugar here? This is deoxyribose. Where do you find deoxyribose? DNA. That's the DNA sugar. In RNA, you have ribose. So we'll end up talking about sugars. But they like to be in the chair conformation. And why? Because this minimizes the torsional strain. We still, every one of these have the 109.5 degrees, the sp3, that's all good. But what's different between these different conformations are the hydrogens. So if you look at the Newman projections, everything in the chair, are good. The problem with eclipsing hydrogens in the flagpole hydrogens. So you need to see that in the boat here, okay, you have a hydrogen here and a hydrogen here, okay? And these are called flagpole hydrogens, and that's why they're higher energy, okay, the flagpole hydrogens. But in order to go to this lower ring flip, you have to go through the half chair and the twist boat. All right, now let's go to page 174 and 175 of your book, and it's all about how to draw the chair. So we're going to practice how to draw the chair, and that's what we'll be doing in class. Okay, so page 174 and 175, I'm sure you have a slide, and this is how to draw the chair conformation of cyclohexane. Okay, so step one, you want to draw your, you remember that when you were learning how to draw your letters in elementary, okay? So you want to kind of draw your little paper like that, and then you want to draw these two are going to be parallel. Let's see, let me just draw. That goes there, okay? And these are parallel, and then these are parallel. Okay? So you see how the blues are parallel? They're 1, 2, 3 over. The reds are parallel, and the greens are parallel. And then you can go in here and you can erase this if you want to be like a pro. But you see how it has a little bit of slant to it? Okay, so you have a little bit of slant. All right, and then that's how you draw that. And now what you want to do is the flip. We can draw our other one. Okay, so this goes through a flip. So you see how this one's up here? Now it's got to be down here. And then this one here has to be up here. So to do the flip, you go up like that. And then the red goes to kind of to the middle of the page. So let's see. We'll do the green, red, and then red here, the green there, and then the blue is supposed to be parallel, and you want a little bit of a slant, okay? And it's not always going to be perfect, and it's okay. It doesn't have to be perfect, okay? That's not what I'm after. What I am wanting is for you to try, and as long as you have practiced a little bit, I can tell. So I need a little bit of practice. You can always go back and clean it up a little bit. Okay, so you need a little bit of practice. Now, what I do need to be able to see is a little bit of slant, because the first thing you do, step one, is you draw your framework. Okay, so that's what you just did. You draw your framework. Step two, and I want you to always do this, put in all axial hydrogens. They alternate up, down. Sometimes I just give you a chair and you have to label. So you see how this is pointing down? You draw your hydrogen down. Then this goes up, and you do up. When you do axial, they need to be perpendicular. They need to be in the 90 degrees, okay? If they're not, then I get upset, because remember how I told you this is a drawing class. So you see that's up, down, up, down. This will be very important. This one's down. See right here, it's up, down. Now you go up, so it's up, down, up, down, up. And I have no sympathy when I'm grading these, because I will be grading 20-some students' papers. And so I'll be grading a lot of these. For two of these, I will be grading like 40-some. And you are only in charge of drawing two. So I think that you can do your two correctly when I have to check each one of these and go up, down, up, down, up, down, up, down. Okay? So step two, you put in all your axial hydrogens, and they're called axial hydrogens. And you'll even label them axial. Okay? So a lot of times when you go through here, you'll say, okay, this is axial down. This is axial down. This is axial up. Okay? Axial up. Now, the reason why you want to do that is you want to be able to come in step three. Now you can put in your equatorial hydrogens. And when you do this, and when we start doing disubstituted compounds, you can erase one of the hydrogens and put in like a methyl group or an isopropyl. Okay, so these are E. Okay, E for equatorial. Now equatorial, so if this is down, this hydrogen has to be equatorial up. So that's why you want to do your axial first. This is axial up, so this needs to be equatorial down. Do not put it inside the ring, okay? Make these go out. This is axial down, therefore this is equatorial up. This is axial up, therefore this is equatorial down. And you notice these have a little equatorial. They're not axial. This is axial down, equatorial up. Axial up, equatorial down. Okay, it's the same here. Axial down, you pick any one you want to start. Equatorial up. Axial up, equatorial down. Axial down, equatorial up. Axial up, equatorial down. Axial down, equatorial up. Axial up, equatorial down. Okay, and that is how you draw and label axial equatorial positions. Okay, now the next thing is your monosubstituted. So what if you have this compound? What if I asked you to draw the chair conformation of what is this compound? Methyl, cyclohexane, and do the flip. Okay, so you would have to draw this. So you draw your framework, and then the flip. This now is down has to be up, and this corner here that's up has to be down. So then you draw like that. All right. Now you put your methyl in here. If you want to put your methyl CH3 up, okay, now it's on the blue. So that is axial up. Okay, because if you put in your hydrogens, you could go and put your hydrogens in here for each one of them. Okay, and this would be axial down, up, down, up, down, up. Okay, and then you come here, and then if you put hydrogens, you go down, up, down, up, down, up, and then this one here is on the blue, and so now it becomes CH3 equatorial up. So when you do the ring flip with your substituents, this is the rule, okay? So if you want to do step 4, this would be step 4 on the ring flip. So we're doing a ring flip. The rule is up stays up, down stays down. Now in class I make everybody chant that. Equatorial becomes axial, axial becomes equatorial. So this is axial up, and this became equatorial up. Did up stay up? Axial become equatorial? And so that's the ring flip. And then what you have to do, and this is what we'll be looking at, is which one's the most stable conformation. It would be where the methyls in the equatorial. So then your equilibrium arrows would not be like that. Your equilibrium arrows would be more like this. And most of the time at room temperature, probably like 90%, you're going to have the methyl group. It's kind of bulky. You want it in the equatorial position. And why? Because here you have this nice little sterics right here, okay? So you have these sterics here. And this is called, what's this called? This is called gauche. Okay, and it's a steric dihedral angle interaction with this hydrogen here. Because remember, the CH3 is going around and around and around and around. Okay, it's rotating around that bond. Here, it can rotate, and you don't have, this has no gauche steric interaction. Okay, and so you can also see that on page 177 in your book, where the Newman projections are drawn, and it's showing you that you're more likely going to have that methyl group in the equatorial position. Okay, so then what about disubstituted? Okay, so let's go to another slide. Okay, so if you have something like this, and you might get something like this methyl, okay, or, okay, so let's name these. Which one? So you might get which cyclohexane is the most stable. And you might have to explain that. And when you explain, you're going to have to draw the chairs and the ring flip. Okay, so that's how you explain that. So first, let's name these compounds. So 1,2-dimethyl cyclohexane. This one's 1,2-dimethyl cyclohexane. They both have the same molecular formulas and the same connectivity, except what? They're both 1,2-dimethyl cyclohexane. Orientation in space. So the first one is trans, the second one is cis. So these are stereoisomers. All right, so which one is more stable? Well, let's draw the chair, the first one. Step 1, you draw your cyclohexane ring. Step 2, you put in your hydrogens. So that way you know you're up, down. Okay, so up, up, down. Okay, I'm going to do the same thing here. I'm going to draw my chair. Now, if I draw this a lot quicker than you, it's because I've been doing it longer than you. Okay, I've been drawing this longer than you've been alive. All right, now we put in our CH3. Okay, so I want to put it, this is coming at me, so I'm just going to go ahead and put that up, and I'm going to make that the carbon. I'm going to pick that one. I'm going to put CH3, this is equatorial up, because I know I want it equatorial. Okay, well then this CH3 has to be next to it. I can go back there, or I can go here. So if I go here, it has to be opposite. So if this is, if I made this up, this has to be down. Okay, so then this is going to go here, and it's going to go down, and that's equatorial down. Okay, both substituents are equatorial. Okay, that makes me happy. That's going to be energetically favorable. Okay, so if I come over here and I draw this one, okay, so I go with the methyl up, so I'll make that one here, CH3, that's equatorial up. And then this CH3 has to be also up. So now I'm just going to erase my methyl here, and I'm going to put the CH3 here. That's axial up, because they both have to be up. They're on the same side. Okay, so now I have one substituent in the axial and one equatorial. So this one's not going to be as stable. It's having both substituents in the equatorial position. And when you're drawing compounds, like so if you have something like an isopropyl is extremely bulky, okay? So isopropyl will always be in the equatorial. Also tert-butyl. So if you have a tert-butyl, okay? A tert-butyl, those groups, sometimes they're called locking groups, okay? And you'll want to, so if you have to choose between which, you see these are both methyls, right? But if you had to choose between which one gets the equatorial position, you would have to choose the larger group. Could you convert this into the two-dimensional? Okay, so this would be going down, okay, carbon number 1, and then carbon number 2 here, the methyl would be going down, okay, so that's how you would convert that. Those represent the same, and you want to be able to do stuff like that. So here you have the isopropyl, or tert-butyl going down, there you go. That is actually not drawn correctly because we have too many carbons. Make sure you do that. There's one carbon, 2, 3, 4. There's your tert-butyl. It's going down. 1, 2, 3, 4. 1, 2, 3, 4. It doesn't really matter where you start. Now this methyl here is going down. So if you want to do down as your dashes, that works. All right, at this point, I think we're ready to work our Pogel exercise 5B. Okay, so 5B, we have a couple models. We have, looks like 4 models. So we're going to go ahead and do this. Model 5B, or Pogel 5B, Model 1. Okay. Let me redraw this one. And you see that we have this here and the dotted line here, dotted line here. This is the boat. This is your carbon. This is this carbon here. We'll just put a, and this is called the chair. All right, now these are conformations of cyclohexane, and it's a non-planar conformation with bond angles close to 109.5. The two main conformations of cyclohexane are shown, the chair and the boat. Okay, let's answer question 1. What are the names of the two main conformations of cyclohexane? Chair and boat. The chair conformation can be envisioned as being two triangles. So you see the two triangles that are connected by two parallel lines as shown above. Explain how the arrow shows conversion from the chair to the boat. Okay, and I didn't draw this in here, but that arrow, the bottom carbon, see the red carbon, actually flips to the top. All right, so you're saying here you need to make a model kit. Use your model kit. You can. This would be a good thing for you to do, and then rotate this and see if you can actually flip with your model. Once your group reaches consensus on the above questions, predict whether the boat or the chair conformation is the most stable. Okay, and if you did that model, you would see that the chair is the more stable, and the reason why is because we have no 1,3 diaxial interactions. Okay, look that up. 1,3 diaxial interactions in your book and read about that and look at your slides on that. Okay, model 2. This is the axial and equatorial. And I'm kind of going fast through this because we've already done this, right? So remember how I told you to draw your chair, your framework. So you draw your framework, and then you draw in your axial up, axial down, up, down, up, down. And then you want to do equatorial, and this is 1, and then this is 2, equatorial up, okay, so we have our nice little model 2 drawn. So now let's answer our questions. So turning the page to page 60, let's look at question number 6. How many axial hydrogens are on carbon 1? So this is carbon 1. How many axial? 1. How many equatorial? 1. How many axial hydrogens are on carbon 2? Axial, 1. How many equatorial? 1. Based on your answers to A and B, what can you conclude about the number of axial and equatorial positions at each carbon? I wrote there are 1 axial hydrogen and 1 equatorial hydrogen on each carbon. Describe how to locate the axial position with reference to the plane of the ring. Okay, so you see how axial goes up, down, up, down, up, down. So in the plane of the ring, it is perpendicular. You remember how you do, that's the sign in geometry for perpendicular, and they alternate. Okay, that's important, that they're going to alternate up and down. E, describe how to locate the equatorial position with reference to the plane. So first, you got to draw in your perpendicular axial, okay? Because that gives you your orientation. And then you can say if this is axial down, this must be equatorial up, and you try to draw it as parallel to this is here. It's supposed to be parallel to like 3 over. So like this here is parallel to this here. So that's how you know you're drawing. So if you come here, this blue should be parallel to here, you see. So it's kind of hard to get it exactly right, but that's the way the orientation is. Okay, label each of the remaining hydrogens in the cyclohexane as axial or equatorial. And I'll say go ahead and do like this. This is axial up. Do this one is equatorial down. This is axial down. Label this as equatorial up on your paper. This is axial up. Draw in equatorial down. Axial down. Equatorial up. This one's axial up. This is equatorial down. And you want to be able to label those. All right, so we're going to go on to model 3. This is the chair interconversion flip. Rotation around the single bonds of cyclohexane results in the conversion between chair conformations. Okay, so if we draw the chair here, and I'm really not happy with that right there. Okay, so this is chair 1, and then it goes through the boat. Okay, there's my boat. And I'm only going to ask you to draw the chairs with the ring flips in your open response questions, okay? All right, so then this is chair 2. And then we're going to label this. We have the axial B, equatorial A, and that's carbon number 1. 1, 2, 3, 4, 5, 6. And then here, this becomes, make that A and that's B. A. And this is B. 1, 2, 3, 4, 5, 6. And then we have A, B, and we have 1, 2, 3, 4, 5, 6. All right, now let's answer our questions. 11. In chair 1, group A is, what would that be labeled? Equatorial up. So E up. So equatorial. Equatorial. In 12. In chair 2, group A is axial up, right? Axial. So you can circle that. What conclusion can be made regarding the orientation of a group when a ring flips? Up stays up. Equatorial becomes axial. Number 14. Consider group B in both chair 1 and 2. Does the conclusion you derived in question 13 agree with the orientation? Well, what happened to B? B in chair 1 is axial down, and in chair 2 it is equatorial down. So did down stay down? Yes. And did axial become equatorial? Yes. Okay. Once your group reaches consensus on the above questions, predict whether there would be any energy difference between chair 1 and 2. Okay. If they're both hydrogen, no difference. Okay. The only time there's a difference, and we kind of touched base on this. If you have bulky groups, so if you're comparing a hydrogen versus a methyl, or methyl versus an ethyl, okay, those are going to, the bulky groups need to be equatorial. Otherwise, you're going to have that 1,3 diaxial gauche steric interaction, okay, and that's not energetically favorable. Oh, we have more questions. Okay, 16. On the left below is one chair conformation with methyl cyclohexane. So we do the ring flip. All right, so let's try to do this one. You all can try to answer questions while I draw. This is the chair flip. Okay, now 16A. Identify whether the methyl group is in the axial or the equatorial position. So this methyl group, it's axial, right? Axial up. B. Complete the drawing on the right showing the structure that is formed after the chair has flipped. All right, so it's on carbon number 1. And what's our rule? Up stays up. You want to go ahead and draw in your, always draw in your axial. So that's axial down. This is equatorial down. And then the methyl has to stay up. And it's equatorial up. And so axial up became equatorial up. Identify whether the methyl group in the structure on the right is axial or equatorial. It's equatorial, right? All right, number 17. Of the two chair forms of methyl cyclohexane, construct an explanation for why the conformation with the methyl group in the equatorial position is more stable than the conformation with the methyl in the axial. Okay, so this one's more stable. Why? Because the bulky methyl group here, sterics, needs more space, and this does not have the carbon 1,3 diaxial interaction. So like carbon number 1, 2, 3, this hydrogen here. If you made a model, you would see that as this group here is rotating, it would be clashing with that hydrogen there that's at the 1,3. So this one has 1,3 diaxial gauche interactions. This is a steric. You'll find that most things are either steric driven or electronic. So here, when it's in the equatorial, you do not have these steric interactions. All right, now we are on to our last model for this POGIL exercise, and this is model 4. And these are called di-substituted cyclohexanes. And so I will draw these as you can read the questions and ponder. And try to answer the questions by yourself, and then you can go back and listen to me. See if your thinking is correct. That would be the best way to use these videos. All right, these are spatial relationships between two substituents on different carbons in a ring must be identified as either cis which means the same, or trans, which means opposite. Okay, so question number 18. Label each dimethyl cyclohexane as either cis, trans, or neither. So what did you do with the first one? Cis. This one's neither. Okay. This is trans, and this is trans. All right. Consider the chair conformation, number 19, for the 1,2-dimethyl cyclohexane on the left. So I will draw my chair as you answer the questions. All right, and it says for 19A, the two methyl groups are, okay, so this methyl group and this methyl group. What are they? I like to label things. So you have axial up, and this is axial down. Up and down makes them trans. B. Draw the methyl groups at carbons 1 and 2 after the chair has flipped. All right, so at carbon number 1, this hydrogen is equatorial down, becomes axial down. I always want to do my axial in first, and then this would be CH3. This is equatorial up. So do we follow the rule? Axial became equatorial, up stayed up. Okay, so we're good there. And that's carbon number 1. Carbon number 2 here, we're going to draw in, this is axial down. And here, this one's going to be axial up. And then, so this is CH3, this is equatorial down. Down stayed down. Axial went to equatorial. So yes, we've drawn it correctly. All right, so C, the following statement is true or false. When a chair flips, the relationship between the two substituents remains the same. Explain your answer. Okay, I said true. Both methyls remained on the original side. Okay, so if you look at this molecule is trans. So if you named it 1 trans 1,2 dimethyl cyclohexane, here this one's down, this one's up. This is trans 1,2 dimethyl cyclohexane. Okay, so that's why I said true. All right, your last question for this model is 20. Draw the two chair conformations for trans 1,4 dimethyl cyclohexane. Which conformation would be the most stable? And you need to explain. All right, so let's do that one. What I like for you to do when you're doing your problems is to first draw the two-dimensional compound that you're going to do. So trans 1,4 for dimethyl would be 1, 2, 3, 4. So I first want to see the two-dimensional. It's called two-dimensional drawing with the dashes and wedges. And then I would want you to do your conformation. Okay, and so you can go ahead and draw the ring flip, okay, the chair flip. And so if this is 1, 2, 3, 4, 5, 6. Then you can draw this one just the same. Now, this is number 4, 5, 6. This one's 1, 2, 3, and there's your 1. Then you want to draw in your hydrogens. Up, down, up, down, up, down, down, up, down, up, down, up. Okay, and so we do this first. So we did our framework, we did our axial, and now we have to put in our groups. So let's make this one green, and then we'll make this one yellow. Okay, so let's do this one with 1, 2, 3, 4, 5, 6. So 1 is here, and I'm going to put that as equatorial. That might be challenging for you. Let's make it easier for you. I'm going to change the numbering. Okay, so I'm going to make this 1, 2, 3, 4, 5, 6, and I'll change the numbering here. It doesn't matter. I'm just for teaching purposes. I think this will make it easier for your orientation. Excuse me. All right. So what do we call this one? This one's 6, 5, 4, 3, 2, 1. All right, so the green, we want to make that equatorial up. And then over here, the yellow would be 1, 2, 3, 1, 2, 3, 4, and that's going to be, oh, and you don't do the, sorry, you don't do the dashes and the wedges on this, so this would just be like that, so don't do that. And this would be CH3 down. This is equatorial down. Okay, so this is important. So this is down, and this is up, if you want to keep it coming at you. Okay, so this one's up, and their relationship 1, 2, 3, 4, 1, 2, 3, 4. Okay, so the ring flip then for here is on the 1, the green, and if this is equatorial up, then this has to be axial up. So you can just erase that hydrogen you've already got there, and now you make it CH3. So that's axial up, and then on 4, 1, 2, 3, 4, that's going to be equatorial down. Okay, so this is CH3. This is axial down. So when you look at this, did equatorial become axial? Did down stay down? Yes. And then you do your check here on the green, methyl. Equatorial up. Did equatorial become axial? Yes. Did up stay up? Yes. And then you make sure the relationship is a 1, 2, 3, 4, 1, 2, 3, 4. Okay, so as long as that relationship, it doesn't matter where you orient this. All right, now, so we drew both of them, just like it asked, and then asked which one's more stable. Okay, why did you pick this one to be more stable, the first one? I always draw the most stable. So the methyl groups are both equatorial. So a lot of times you just count, okay, we got 2 equatorials and they're the same size. Okay, the problem with here is you would have this is a 1,3 diaxial gauche interaction. And the 1,3 is the carbon, 1 carbon, 2 carbon, 3 carbon. So it doesn't matter how you number these, it's just saying that 3 away. So here you have another 1,3 1, 2, 3. You have that 1,3 diaxial gauge interaction, and you don't have that here. You don't have this here. Okay, so that finishes up our models for POGIL 5B. It looks like on page 63 you have additional questions. It looks like you get to do exactly what we just did in 21. So draw your two-dimensional with your dashes and wedges, and then draw your ring flip. And I want to see that for 22. So I need to see your, for 22, you can just draw the most stable, and then you can draw your two-dimensional drawing. Okay. 23 is glucose. I'll just go ahead and tell you, when you draw glucose in the most stable for number 23, you want to put every OH, every carbon has an OH in glucose. You want to put each one of those in the equatorial position. Okay, so I'll just go ahead and tell you to draw that. And that will take care of your POGIL activities for today.